everyone. Welcome to the latest Army of Women Research Results webinar. We thank all of you for joining us today. We have a packed audience. We're very, very excited. My name is Leah Wilcox. I'm the Army of Women Research Project Manager, and I have with us Naz Sykes, our Executive Director at the Dr. Susan Love Research Foundation. And we are very excited to have with us today Dr. Harry Ostrer, who worked with us on the Jewish women's breast and ovarian cancer genetic study, and the Army of Women helped him recruit women for his study. And you guys, a lot of you on the call today participated in the study or signed up to be screened for it. So we're very excited to have Dr. Ostrer present to you today about the results of the study. I just wanted to give a brief introduction about Dr. Ostrer. He is a medical geneticist who investigates the genetic basis of common and rare disorders, and in the diagnostic laboratory translates the findings of genetic discoveries into tests that can be used to identify people's risks for having a disease prior to its occurrence or for predicting its outcome once it has occurred. He studies the genetic basis of prostate and breast cancer and adverse outcomes associated with their treatment. He also studies the genetic basis for disorders of sex development and other rare conditions, having recently identified the role of the single transducing MAP kinase pathway in gonadal development. Dr. Ostrer is a longtime investigator of the genetics of the Jewish people. For over 30 years, he has studied the genetic basis of single gene disorders in Jewish populations and implemented new genetic tests and screening programs to benefit Jewish people. He is also an investigator of the genetics of Hispanic and Latino people, including Puerto Ricans, Dominicans, Ecuadorians, and Mexicans who currently reside in New York City. And so as I mentioned today, Dr. Ostrer will be discussing results from his Army of Women study, the Jewish Women's Breast and Ovarian Cancer Genetics Study. Dr. Ostrer, please go ahead.
Great. Thank you very much for that nice introduction, Leah. So I'm very happy to be talking about the results from the Jewish Women's Breast and Ovarian Cancer Genetic Study. And as you'll see, this study really would not have been possible without the involvement of the Dr. Susan Love Research Foundation and the many participants who identified themselves through the foundation and contacted us for participation. So the goals of this study were to identify new genes that convey risk of developing breast and ovarian cancer among Ashkenazi Jewish women, and also to identify modifier genes that reduce a woman's risk for developing cancer despite her inheriting a mutation in BRCA1 or 2. So a modifier gene, as the name would suggest, is a gene that acts on a major gene, such as BRCA1 or 2, to either increase or decrease the effects that are being produced by that major gene. So this will give you a sense of what the recruitment strategy was in order to reach participants in the study. Through the Army of Women, an email blast was sent, and attached to this was a brief screening questionnaire that we used to determine eligibility for participation in the study. At the time, I was at NYU School of Medicine, and the last year I moved to Albert Einstein College of Medicine, but you'll see that there are a lot of NYU links on the slide, and indeed I continue to work along with my various collaborators at NYU on these studies. So a list of potential participants was sent to the NYU study coordinator, and in turn she sent a detailed, secure online eligibility survey. And the participant was notified of her eligibility at the end of the survey. Now, based on this eligibility, the participants were consented via telephone, a study kit was sent via mail, and enrollment was completed upon delivery of the kit back to NYU. And this was really a remarkable way of doing recruitment into a study. As you'll see, we recruited over 1,000 people, and there have been few models of this type before. I have to say that both we and the staff of the Army of Women were rather impressed by how successfully this worked. So we had varying categories of women who were eligible for the study, and these included women who had a family history of breast cancer, but it couldn't be demonstrated on the basis of a mutation in the BRCA1 or 2 gene, or they had been untested. So these women were characterized as being high risk. There was a second group of BRCA1 and 2 mutation carriers, but who hadn't developed cancer by the age of 60. There was a third group of BRCA1 and 2 mutation carriers with breast and or ovarian cancer. And then there was a group of women who were 70 or older, but without a history of breast or ovarian cancer by the age of 70, and who did not meet the high-risk criteria for entry into the study. But nonetheless, for studies of this type, it's important to have a so-called control population to compare with. So indeed, we were looking to compare women who came from high-risk families, and as you'll see in the initial study, did not have a demonstrable mutation in BRCA1 and 2, against age-matched women who had the population-based risk for breast cancer. So we recruited 1,187 participants altogether, and this represented 54% of the total number of participants that we enrolled in our study. So in the initial study that I'm going to report, we performed a so-called genome-wide association study. And this has become a rather popular means for looking at genes that convey risk for a variety of different disorders. On the right-hand side of this slide is a summary of so-called GWAS studies through the second quarter of 2011. And it shows that 1,449 studies had been published, which had a high degree of significance, a so-called statistical p-value of less than 5 times 10 to the minus 8th. And these had been performed for 237 different traits, which included breast cancer and other forms of cancer, looked at other diseases, also looked at other traits such as responses to drugs or development of certain types of toxicity following treatment with drugs or radiation. So a genome-wide association study is a way of scanning the entire genome, the entire DNA content for disease-causing associated genetic variants. These can detect small genetic changes that are referred to as single nucleotide polymorphisms. These look at single changes in the DNA code, for instance, a change of the base A to T. For these types of studies to be successful, there's a need to recruit large populations to be able to detect significant associations. And these studies have to account for population as well as phenotypic variants. So risks that may exist, for instance, in the Ashkenazi Jewish population, as you'll see, may not be identical, for instance, to those that are found in the larger European American population. And there can also be variation for one particular form of the disease versus another. So for instance, the types of risks that are being conveyed for breast cancer may in fact be different from those that are being conveyed for ovarian cancer. And these variants can provide useful information in so-called personalized medicine, that is developing individualized treatments or individualized models for predicting disease risk as well as predicting responses to treatment. So to give you an idea of the types of tools that we have for predicting breast cancer risk, since the late 1980s, there's been a tool that's called the Breast Cancer Risk Assessment Tool that includes information about age at first birth, age at menarche, number of breast biopsies, and race and ethnicity. And indeed, in the course of our recruiting subjects into our study, we asked them questions that included these bits of information that were included in this risk assessment tool. And typically, for women, the general population risk is on the order of 12% for developing breast cancer. And if they have a first-degree or second-degree relative with breast cancer, then their risk is going to be increased. The degree to which it's increased is going to be determined by the number of affected relatives. Now, typically that serves as a trigger for women to have genetic testing for BRCA1 and 2 mutations. And if a woman is tested and is found to have a BRCA1 or 2 mutation, then her lifetime risk for developing breast cancer is going to be on the order of 80%. But if she is negative for having a BRCA1 or 2 mutation, then her risk doesn't revert back to the population-based risk. It's the same as anyone else who has one or more first or second degree relatives for breast cancer. But we don't know exactly how to counsel them. Now, this becomes important because for women who do have these increased risks, we will suggest that they should be closely monitored and or that they may want to have some sort of preventive treatment that may even include having surgical removal of their ovaries or potentially even their breasts. But as I mentioned, for women who have no BRCA1 or 2 mutation, we don't know exactly what to say to them about the risk. Therefore, we think the tools of genetics can be used to improve this risk assessment. Now, in the course of designing this study, we decided to focus on the Ashkenazi Jewish population. This is a group of 5.5 to 6 million people in the United States. These people share not only the Jewish religion, but also ancestry. Typically, all of their grandparents came from Eastern Europe. And we've actually shown in another unrelated study that their degree of relatedness is what one would expect for 4th to 5th cousins. Now, overall, this group makes up 2% of the U.S. population, and I like to think of them as America's largest genetic isolate. And by studying Ashkenazi Jews, it reduces some of the complexity. It says here, major confounding effect of population stratification. That is, it eliminates the possibility of mixing risks that may be found in different population groups. Now, in much earlier studies that we and others did in this population, we found that about 2.5% of Ashkenazi Jewish women, and for that matter Ashkenazi Jewish men, carry a BRCA1 or 2 mutation compared to 0.1 to 0.3% of the general population. Now, on the basis of explaining risk for breast cancer, this accounts for about 20% in high-risk families, but it means that for the remaining 80% of high-risk families, that there are other genetic factors that are operating, and as we've seen, in order to provide better genetic counseling to these families and individuals, we need to have new genetic risk assessment tools. So in this study, we included 3 different cohorts of Ashkenazi Jewish women who were negative for BRCA1 and 2 mutations. We analyzed so-called germline DNA that came from their blood or saliva, and we studied them on high-density gene chips, or so-called SNP arrays, to look at least 500,000 different sites across the whole genome. And this gives you a sense of what these 3 different cohorts were. The first had been identified by Bert Gold and his collaborators and published in the journal PNAS in 2008. And this included 3 or more affected relatives in a single lineage, and their age of onset was 51. The second cohort came from the Breast Cancer Family Registry and had 1 or more affected first or second degree relatives, and their typical age of onset was 47.5. And the third cohort was ours, that included 3 or more affected relatives in a single lineage with a typical age of onset of 52.2 years. And in each group, there was a control cohort of women who did not have breast cancer, who had no family histories, and this gives you a sense of what the average age of enrollment was within each of these groups. So this shows that in cohort 1 there were 239 cases and 294 controls, and this was matched against cohort 2, which was pretty much similarly sized with 238 cases and 230 controls. And in overlapping between those 2 cohorts was 101 significant SNPs, 101 significant markers. Now, some of these may be true positives and some may be false positives. So we included our third cohort as well of 203 cases and 263 controls, and overlapping within these 3 cohorts was 7 SNPs. Now, when we built a model to explain how these might predict the risk for breast cancer, we observed that these 7 SNPs significantly contributed to the breast cancer risk in Ashkenazi Jewish women who were BRCA1 and 2 negative. And these SNPs were not previously linked to breast cancer risk because most of the studies of this type that have been performed in the past have been done in European or European American women who are genetically different from Ashkenazi Jewish women. And from this, a model was built that looked at how well these SNPs were able to perform in predicting the risk. So this is a so-called receiver operator or ROC curve, and if it is there is a perfect tool, then it will be completely sensitive. It will fall on this axis here, and it will be completely specific as well. There will be no false negatives and no false positives with this test. And if the predictive model is completely random, then it will fall on this line here. This conveys the so-called area under the curve of 50%, whereas for a perfectly predictive test, the area under the curve is 100%. There's nothing that is outside the curve. Now, as you can see, for our particular model, the area under the curve was about 74%, which is actually quite good. Typically, the type of predictive tools that doctors like to use have an area under the curve, an area under the receiver-operator curve of 80% or greater. So we took away from this that defining these genetic variants can help us to better personalize our breast cancer risk assessment for those women who come from high-risk families. But we thought that we shouldn't stop there because we'd like to have tools that exceed this threshold of 80% under the ROC curve. So in summary, you can see that we've identified new SNPs that are associated with familial breast cancer risk in Ashkenazi Jewish women without BRCA1 and 2 mutations, and that we've developed a risk model that can help predict breast cancer risk more accurately within the Ashkenazi Jewish population. Now, as many people know, we live in quite exciting times in human genetics, and for instance, it is now possible for us to sequence large regions of human genomes quite accurately and at a relatively low cost. And so a tool that we have taken now is the method of whole exome sequencing. And this looks at regions of the genome that are expressed rather than the entire genome, thus reducing the complexity and the cost. So these are the so-called coding regions or exons of the genome, and they make up about 3% of the total genome. By applying the strategy to a well-defined high-risk group, such as Ashkenazi Jewish women who come from high-risk breast cancer families, we can identify genetic changes that predispose people within the population to an increased risk of disease. And we're applying this to high-risk groups, including those who have multiple family members affected with breast cancer, those with early onset breast cancer but no family history, as well as groups that have well-defined, that come from well-defined populations and are likely to have common founder mutations such as Ashkenazi Jews. And this method is being used now to identify new high-risk cancer variants in a study that we have that's called the Genomics of Breast Cancer Risk. Now, in this study, we are defining the genes and the causal variants in unique families with multiple cases of breast and ovarian cancer where the genes are unknown by using whole exome sequencing. We're defining the genes and the variants within those genes that underlie early onset breast cancer with no family history, and we're determining the prevalence of mutation in these genes in the high-risk breast cancer population. So I think this gives you an idea of the work that we've done and the type of work that we are continuing to do. And we're most grateful to all of you who are participating in this webinar, who have also participated in our study. And of course, we're most appreciative to the Dr. Susan Love Research Foundation Army of Women for their assistance with making the study possible. In the slide, if you are not a member of the Army of Women, here's how you can sign up in order to become involved. And also in the current tab, you can get a listing of current Army of Women studies. So thank you very much for listening today and for your support. And I'd be happy to entertain any questions.
Thank you, Dr. Ostrer. That was wonderful. And we do have some questions. And I think we'll start with a general question from Lynn. She asked if you could give a definition of SNPs again and kind of clarify what SNPs are.
Certainly. So a SNP is a so-called single nucleotide polymorphism. And in the type of study design that we used with gene chips, these represent milestones on the gene chip array that enable us to look not only at that milestone, but also at the surrounding regions as well. So these SNPs serve as tags that provide a basis for identifying the risk. It's important to state that we don't know whether those individual SNPs convey the risk or whether it's a SNP nearby that may be conveying the risk. And that's part of the reason why strategies such as genomic sequencing are now entering into genetic research in order to be able to say this is what was causal and this is what was a marker for causality, but not directly causal itself.
Great, great, thank you. All right, so we have a question that was sent in prior to the webinar. Deborah wants to know what is the state of research on treating or counteracting the dangers caused by carrying a defective BRCA gene? What approaches are they looking at? She's curious because the gene is in her son-in-law's Lutheran family, not her Jewish one, and she wonders what the prospects are for her 3 granddaughters should they inherit it from their dad.
Sure. There are a variety of different strategies that are used to mitigate the risk of breast cancer, and these include screening using techniques such as mammography and ultrasound and MRI. This isn't typically performed in very young women. Therefore, we don't recommend that women before their late 20s typically have genetic testing and start receiving surveillance for breast cancer. But in many instances, if a breast cancer is identified early, well, then it can be successfully treated. As women have gone through their age of childbearing, if they are a BRCA1 or 2 carrier, they may want to consider having their ovaries removed. Now, this, of course, is a major decision, but removing one's ovaries reduces the risk of ovarian cancer in a BRCA1 or 2 carrier by about 95% and reduces the risk of breast cancer about 50%. And some women choose to have their breasts removed again at some time in adulthood because that also reduces their risk for breast cancer on the order of 95% or more. These are decisions that obviously are not made lightly. Now, it's been shown as well that certain types of drugs can be useful if perhaps for a limited time for reducing the risk of breast cancer. Most notably among these is the use of the anti-estrogen drug tamoxifen.
All right, thank you, wonderful. So Nancy has a question. She wants to know if you found that the Jewish women you tested, did they have more or less gene mutation for breast cancer? I'm assuming she just means then in the general population.
There certainly is a higher frequency of founder mutations for BRCA1 and 2, and that's been known for some time. In the larger European American population, there are over 40 variants now that have been identified that convey risk for breast cancer. We found that some of these conveyed risk for breast cancer among Ashkenazi Jewish women. I didn't show the result of that work. And then in addition to that, there are these 7 new variants that were found.
Okay, wonderful. So Terry has a question. She says that she has a BRCA1 mutation, as does her sister. Terry has had breast cancer and her sister has not. Terry has 2 sons, aged 31 and 34. Her older son went for genetic counseling and was advised not to be checked unless he has daughters or to wait until he is 40 or older. Her sister has a daughter who has been checked and was negative. How important is it for Terry's sons to have a genetic test?
The major risks that are associated with BRCA1 and 2 mutations are for breast and ovarian cancer. And for men who carry BRCA2 mutations, there is an increased risk for developing breast cancer. So that may in fact be important to know. Men who are BRCA2 mutation carriers may want to engage in a program of increased surveillance for breast cancer. For BRCA1 mutation carriers, there is a slightly increased risk of colon and prostate cancer, but it's not of the sort of magnitude that a man, for instance, would start engaging in some sort of increased surveillance program during his 30s. But with these types of individual family situations, it's always important to caution that people should get individualized genetic counseling from their local genetic center to make sure that the risks are being assessed correctly, and that, for instance, I haven't misheard something and given faulty information.
Wonderful. All right, I hope that answers Terry's question. I'm sure it does. So Deborah has a question about the SNPs. She wants to know how these new SNPs predict risk versus the BART testing that is done in high-risk BRCA1, BRCA2 negative women.
Right. So these convey risk in addition to the BC-RAT or Gale model that we were looking at. So although that is a very popular model, the area under the curve for it is only about 67%. So it's really not a terribly accurate model, although it's been a popularly used model. So the incremental risk that is being conveyed by SNPs, is measurable on top of the BC-RAT model.
Dr. Ostrer, I'm sorry, I put us on mute.
I was saying, gee, it's getting strangely quiet here. When it gets very, when it gets very quiet at my house, I get nervous that there's mischief going on.
Well, Kelly wants to know, how far away do you think we are as far as actually being able to do these tests and have them covered by insurance?
Insurers commonly pay for testing for BRCA1 and 2 mutations. I think that as various forms of sequencing for more genes or for entire exomes becomes popular over the next couple of years, that there will be a press for insurers to pick that up as well. So, I think we're a couple of years away from it, and what the insurers are going to be looking for is that the information proves to be useful for clinical decision making. And I would argue that that's why it's important to have studies such as ours and similar studies performed.
Great. All right, Louise has a couple questions. She wants to know, how does this study apply to late onset cancers, and does it apply to postmenopausal women?
Right. That's a really important question, and we think that the risks that occur in postmenopausal women are different from those that occur in premenopausal women, and that it's going to be important to distinguish what the age of onset was for these tumors that are being studied. So the biology would suggest that postmenopausal breast cancer is a somewhat different disease from premenopausal breast cancer, and that's probably going to be sorted out over the course of these studies, both ours and others.
All right. Okay. Oh, no. Thank you so much. Margaret has a question. Margaret wants to know, would use of exome sequencing cause you to miss mutations in gene expression control regions? Can't mutations in control regions contribute to cancer risk?
Yes, they certainly can. And many of the exome arrays that are used now will include some of those regions. And as some of the listeners may be aware, there have been a major series of papers that have been published in the last week about the so-called ENCODE project, which is an encyclopedia of the organization of different elements in the human genome. And so regions that were previously thought to be comprised of junk have now been shown to have functional significance. So I think that the new exome arrays are going to be including many of these ENCODE elements as well, so they will be picked up. Important question, though.
Great, thank you. We have a question. Louise wants to know, what other tests do you recommend for BRCA negative with first-degree relative with breast cancer?
Right. I think that there may be other genes that have been identified now that that might be tested for, genes such as P53, RAD51C. I would think that before going down this path that it would be important to have personalized genetic counseling to make sure that it's appropriate for that individual.
Okay. Wonderful. So Jill wants to know, what is the difference between BRCA1 and BRCA2?
BRCA1 and BRCA2 are two different genes. They are located on different chromosomes. BRCA1 is on chromosome 17. BRCA2 is located on chromosome 13. Nonetheless, they serve similar functions. They both encode proteins that will associate with one another. And we know, for instance, that they both play a role in the repair of double-stranded breaks that occur in DNA. So they're different genes on different chromosomes, but they have similar functions.
Okay, thank you. Elaine has a question. I think she just needs some clarification. She wants to know if any of the 7 genes that you identified with the SNPs have a known function.
Yes, they do. I don't have them in front of me right now, but off the top of my head, some are genes that play a role in cell signaling and the transmission of cell signals, and so the fact that they were identified as risk genes that may be altering cell signaling doesn't come as a particular surprise.
Great. I think that was a really good question. So Nancy wants to know a couple things. So are you saying that with the family history, is it more important for Ashkenazi Jewish people to be tested?
I think it's important for anyone who has a significant family history to be tested. The major difference between Ashkenazi Jews and other people that we know about, in addition to these 7 risk SNPs, is the increased prevalence of these BRCA1 and 2 founder mutations. Now, it's possible that as a result of our study that we will identify founder mutations in other genes that are prevalent in the Ashkenazi Jewish population, but it's too early to say.
All right. And Nancy follows that up with a question about her husband's side. What about her husband's side of the family passing it on to her granddaughters?
Without knowing specifically about their family history, it's hard to answer that question.
All righty, no problem. Amy has a question. Her family, they're Ashkenazi Jewish. Her 25-year-old daughter is BRCA negative, as is she. Amy had breast cancer, and so did her maternal grandmother. Her twin brother also has essential thrombocytosis, JK2 mutation. What else should she do for monitoring her cancer risk?
Right. I'm not sure that she's necessarily a candidate for increased genetic testing until we have a clearer indication about the utility of genetic testing for the Ashkenazi Jewish population. So again, I think that studies such as ours should contribute to that over the course of the next couple of years.
Excellent. Stephanie wants to know if you're able to tell the audience what the 7 SNPs are, or if they're published and where they could find that information.
The manuscript has been submitted to a journal called Human Genetics. It's now under review, and we hope to hear favorably about that in the next couple of weeks. Once the manuscript has been published, then the information will be in the public domain.
Excellent, and we'd love to share that with our Army.
And of course we will make the manuscript available to Army of Women.
Excellent, wonderful, thank you, thank you. And then, do you want to, Dr. Ostrer, talk a little bit about your next study? And I know that you are hoping to recruit through the Army of Women for that one as well. We did have some people interested in that.
Right, so there, although we are going to be doing exome sequencing in the cohort that we've recruited already through Army of Women, specifically for this issue of identifying additional SNPs and so-called founder mutations. In the new study, we want to recruit people who come from really high-risk families, so 3, 4, 5, 6 affected individuals in a single lineage. Because that represents another way of approaching the identification of risk genes. And so in order to participate in the new study, people have to come from these very high-risk families. It's a strict eligibility criterion, and they can't participate as individuals. They have to have at least one other family member in the same lineage participate as well. So they have to sign up their family members. Ideally, multiple family members will be breast cancer survivors and will be willing to participate in the study. So this will be, if you will, a personalized sequencing study for identification of breast cancer risk within that family.
Excellent. Well, we're very excited to help you out with that next study and share that with the Army of Women. And I would like to say that we do have other questions, but they're a little bit more personal nature on these questions, so if anybody has asked a question and is waiting for a response, what we'll do is we'll answer them separately. We'll share them with Dr. Ostrer via email, and then either I will respond or Dr. Ostrer will respond directly. We'll get that figured out so everybody gets their questions answered. Thank you so much, everyone, for putting your questions in there. And so if anybody has any follow-up questions after the webinar, you can email us. The email is research@armyofwomen.org, and you can email us questions, and we can send those out, send responses out. And then we will also post the webinar, the recording of the webinar, on our website by the end of the week. So if anybody wants to watch it again or share it with your friends and family, we'd love for you to do that. So Dr. Ostrer, thank you so much for joining us today. This was a great presentation. We really appreciate everybody who signed in and listened and asked their questions, and we will look forward to answering those.
Oh, it was my pleasure to participate today, and of course we want to preserve people's confidentiality in all of this, so I guess that's what you need to sort out, Leah, whether people want to be identified to us or whether they want to retain their anonymity and, for instance, have us convey their responses through you or, around you or however that happens to be.
Exactly.
Right.
We'll get in touch with everyone who has asked questions, and we'll get back to you. So thank you, everyone. And again, you can go on to www.armyofwomen.org to look at our current studies, to invite friends to join if you want. And we appreciate everybody's support. Thank you so much.